Cloning strategies. First, briefly we will focus on the introduction or the overview of cloning strategies. In previous topics we discussed that what are the different methods that can be used for cutting and joining DNA molecules, and how the DNA molecules or fragments, they can be ligated with suitable cloning vectors. So any cell-based cloning, it requires 4 basic steps. The first step is that there must be a method that can produce discrete DNA fragments from any source. So for this purpose, restriction endonucleases, they are the most suitable choice. So after DNA fragmentation, the next step is that there must be some reactions available that can join our discrete DNA fragments into a suitable cloning vector. And you have the idea that different cloning vectors are preferred. That may include plasmids, bacteriophage lambda, cosmids, and even bacterial artificial chromosomes or the yeast artificial chromosomes that can clone comparatively larger DNA fragments. They can be used as a cloning vector. So this is the second step, where the target DNA, it is ligated with suitable cloning vector. Third step is that this recombinant cloning vector, it should be introduced into a suitable cloning host where it can be replicated. As far as suitable cloning host is concerned, number of prokaryotic microbes and a few eukaryotic microbes like the yeast, they can be used as a suitable host organism. In addition to that, higher plants or animals, they can also be transformed. So in the host, after transformation, our recombinant vector, it will multiply there. And the final step is that we must have some suitable strategies to detect our cloned gene into the host organism. That is proper. Some screening strategies must be available so that we ensure that our gene of interest, it is present in that particular host organism. This diagram, it gives generalized overview of cloning strategies. As I told you that 4 basic steps are required for cloning. First step is to isolate DNA fragments. And for this purpose, to generate DNA fragments, different techniques are available. The most suitable one is to use restriction endonucleases. So the source DNA, it can be digested with appropriate restriction enzymes to produce discrete DNA fragments. In addition to that, mechanical shearing, it can also be used to produce DNA fragments. And the DNA that is synthesized from the messenger RNA, which is called as the cDNA, it can also be the source of the gene of interest. Another source of DNA is that if we know the sequence of our gene of interest, then it can be chemically synthesized in the lab, and it can also be used for cloning purpose. And finally, the PCR product. By using specific primer, we can amplify our gene of interest, and then we can use that amplified fragments to join with suitable cloning vector. And these arrows indicates the suitable joining vector or technique. In the second step, DNA fragments, they can be joined by homopolymeric tailing. Then if the source DNA, it has been digested with restriction enzymes, then it can also be ligated by the production of cohesive ends, you are well aware. And in addition to that, blunt ends that are produced, they can also be ligated. And different linker molecules, they can also be used to ligate DNA from two different sources, and in addition to that, TA cloning, which is very common in gene cloning procedure, and different kits can be used for this purpose to clone PCR products. So TA cloning, it is also used. In the third step, in the basic cloning, it is introduction into a suitable cloning host. So here the phage DNA, suppose it can be infected, can be used to infect suitable host organism here in the first step, and then the plasmid can be used to transform our recombinant vector into a suitable host organism, and then in vitro packaging it can be used, like cosmids can be used to infect suitable host organism. And the fourth step, that once we have transformed our suitable host organism, then how it can be detected. The first step is that we can perform hybridization technique. The known probe can be used that can detect our specific DNA sequences. Then PCR can be used to detect our gene of interest, and immunochemical technique, protein to protein interaction, and so on. Different techniques can be used to screen our gene of interest into a suitable host organism. These are the four basic steps that are required for cell-based DNA cloning. On the other hand, if we are going to isolate our gene of interest from very complex source like the human genome, then its screening, most of the time it is very difficult, because the fragments that are generated from the human genome, they will be in hundreds of thousands in number, and to find our gene of interest, it is a laborious process. In order to minimize the size of the DNA fragments from complex source, we may adopt different strategies. The first strategy is that we can clone DNA fragments into a suitable cloning vector that can accommodate larger DNA fragments. And here the most suitable choice is bacteriophage lambda vectors that can accommodate around 20 to 23 kilobase foreign DNA. And here the size of the gene library that will be produced, it contains low recombinant vectors that is easy to screen. So first strategy is that to minimize the size of the genomic library. Another strategy is that we can selectively amplify our gene of interest from complex human DNA. So for this purpose, the gene specific primers, they are used, and especially this technique is suitable if we have the idea that this is the gene that we are going to amplify, and specific primers they must be available.